Well, thanks everyone for attending. My name is Bill. I'm the Director of User Services for the GDC, and today I'd like to talk to you about GDC single-cell RNA-seq analysis. We have a couple other presenters who will be joining me soon. I'll let them introduce themselves when their section starts. So just to go over a bit of webinar logistics, this will be recorded. The recording and the slides will be made available soon as it's done being processed and captioned. And any questions you have, please type in the Q&A panel. They will be addressed at the end of the webinar. So to give an agenda, first we're going to be giving a brief overview of the GDC single-cell RNA-seq harmonization workflow. That'll be Zenu, followed by an overview of the available files. After that, Shin will be giving an introduction to the GDC single-cell RNA-seq tool in the portal. And that will be followed by a demo of the single-cell RNA-seq expression API. Any questions that come from the group will be answered at the end. So Zenu, why don't you go ahead?
Thanks, Bill. Good afternoon, everyone. This is Zhenyu. I'm Director of Bioinformatics at the University of Chicago. I'm going to talk about a brief introduction of the harmonization workflows we have. Next slide. Thanks. So the analysis of the 10x genomic single-cell RNA-seq data in the GDC consists of two sequential workflows, as you can see. The first workflow is the alignment and quantification pipeline, mainly based on the 10x tool called scRNA-Seq. It takes the raw FASTQ file input and generates three main output: the alignment BAM, a raw count matrix, and a filtered count matrix. This filter matrix is not a direct output of scRNA-Seq. scRNA-Seq generates a filter matrix, and we have applied additional filtering steps to further remove empty droplets and doublets that result in what we call the high confidence cells as the filter matrix. The second step of the analysis is based on a R package called Seurat. It uses the filter matrix as input and generate three primary output. The first output is we call it analysis TSV. This output contains dimensional reduction results such as PCA, t-SNE, or UMAP. And also it applied the default graph-based clustering on these cells. Those information are later used for visualization. And also a full analysis log in HDF5 format, also sometimes known as the Seurat room file, are also generated. This is a very big file containing all the key parameters, intermediate steps, and metadata during the analysis. And it contains both steps and data, so including all the intermediate and final transform gene expression data. The last file generated out of this pipeline is a differential gene expression TSV, where each cluster is compared against all the other cells to identify marker genes. This is the standard way for users to figure out what each cluster is by looking at its special marker genes, whether it's overexpression or lack of expression. Okay, that's all about the pipelines. I'll hand it back to Bill.
Thanks, Zenu. So just to go over some of the files that are available for single-cell RNA-seq. But before I do that, I just want to let everyone know that what I'm going to present right now is kind of an abridged version of a webinar that we did in 2021. So if you're interested in kind of a more in-depth presentation of the single-cell RNA-seq data that's available, I would recommend going to look at this webinar, and I'm going to show you how to get to that at the end of this one. So starting with the gene expression files, these are available in a tarball called an MEX format, and in this tarball are three different TSVs. The first one are the barcodes, which is just a list of the barcodes for the sample. The second one is the features, which is a list of each gene ID and the gene name. So a shorter gene name that's not the ensemble ID. And the last one is the gene expression matrix. Um, One thing I want to let you know is that if you're using a Mac, unzipping the three internal files may not work in the file browser. Just double clicking them, I know that's how you unzip files in Macs. For some reason, they sometimes don't work, but they do consistently work if you do this command, the gunzip file command in the terminal. So if anybody has any, I'd recommend using the terminal version. The second file format I'd like to talk about is the analysis file. This is another TSV format, and this contains information on clustering and ways to visualize the data. So this includes the cell barcode, the read count, and the gene count for that barcode, the Seurat cluster that it belongs to, and then coordinates for UMAP, TSNE, and PCA visualization. The last file format, differential gene expression, contains information on the differential gene expression between clusters for differentially expressed genes. So the columns here are the cluster, which are just named as like an integer. The gene, the average and fold change, the p-value, the adjusted p-value, the proportion of cells that are in the cluster, and the proportion of cells that are out of the cluster. So that's just a primer on the TSV files. And the last one I want to talk about is the Loom file, the full Seurat analysis log. Now on the right, this is just a snippet from the current portal on how you can get to this data. It's a single cell analysis file that's of format HDF5. This contains the full analysis from Seurat, as it stores the data on disk rather than in memory, and this can be processed. It's a special format, but there are R packages and Python packages that can be used to look around in these files. So the two that come to mind are Loom R and Loom Py, which are an R and Py package respectively. And now Shin is going to demo the tool in the portal. So Shin, I will hand the screen sharing over to you.
Thank you, Bill. Hey, everyone. Let me share my screen. My name is Xinzhou. I'm a assistant member from Central, and we collaborate with the University of Chicago and also the GDC team to build a visualization feature for the GDC data portal, including the single-cell RNA-Seq. To access this app, and a user can go to the analysis center on the GDC portal and then find the single-cell RNA-Seq card. That's I'm highlighting over here, and then click on the play button. So by default, this app will display a table of available single-cell RNA-Seq experiments, and each row is one experiment. We display multiple columns, including case, sample, project ID, primary site, disease type, and then the experiment unique identifier. And to proceed, a user will select any of these, select an experiment of interest, and to visualize the data from that experiment. I would like to point out some of the cases can have multiple experiments. So such as from over here, these three, they're from the same case, and this case has 3 rows. Each row is one experiment from the same case, and all the experiments from the same case are grouped together. And just one more thing, if a a user, if you happen to see there's no experiments listed from this table, you may want to check your cohort builder. You may be using a particular cohort that doesn't have any single cell experiment. That will cause this table to be empty. In that case, you should adjust or delete the cohort, and then so that it can broaden to include cases that with single cell experiment. So I will proceed to use the first case to demo. I will select it. Once I select it, it will switch to this view, which shows the UMAP from this experiment. Before I go to the details of UMAP, I would like to mention, quickly introduce the layout of this plot. We're now at this tab called the plots, where there are additional tabs, such as this one called samples. I can click on it. When I click on it, it will go back to the same single cell RNA-Seq experiment table that the app initially showed. That this way, the user can always go back to the table and select a different sample, select a different single cell RNA-Seq experiment to look at it. And then, so we have additional tabs, which I will discuss in my following demo. So right now it displays this UMAP plot for representing the single cell RNA-Seq gene expression analysis result, where each of the dot is one cell, and the color of the cell represent the Seurat cluster as indicated by the legend on the right. And this legend is interactive in that I can click on one of the item, one of the cluster number 6. I will get the option of either hide or show it only. When I select that, only the red cells from the cluster 6 are shown, and then I can next click on additional cluster to show them, selectively show that, and then I can go back to showing all of them. This map is also zoomable in that I can use my cursor to drag it to pan and zoom, and also to zoom in. So I can also use the magnifier buttons on the left hand to do the zoom in and zoom out. And finally, I can click on the reset button to go back to the default view. There are also on the top, there are also a checkbox for t-SNE and also PCA, such as I can check the t-SNE, and now it will show both UMAP and t-SNE side by side. And also the current identity of the case and sample, they're all indicated on the right hand of these checkboxes, and these names are always indicated when if you switch to other tabs. For the demo, I will only use the UMAP. This app also allows you to investigate the expression level of gene of interest, such that by overlaying the gene expression level of particular gene on top of this t-SNE or UMAP. In order to do that, I will use the toggling tab on the top and then switch to the gene expression tab here. Now this a gene search box shows on top of the UMAP now. Because this is a brain tumor, I will type in EGFR. Now the request may actually take a few seconds because I have just launched this and my web browser caches the request, so it shows up very fast. So now it has retrieved the EGFR gene expression level per cell, and then overlaid that expression level as a color gradient on top of the UMAP. And the redder a cell is, the higher the EGFR expression is. That is also indicated by this color scale here. So this color scale is interactive, and I can click on it and get more options, such as I can use these two controls to set the color stops, to customize the color for the color gradient. Such I can, let me see. such that I can do this. Now it will change the highest expressed cell will become a dark orange. I can also, right now it says automatic is using the maximum, min, and max of EGFR gene expression range. I can change that into percentile and then press enter. Now you see the max now maximum value for the darkest color now changed, and then more cells got the darker color here. I would like to also point out when the gene expression overlay is being used, you can also generate a contour map. To do that, you can open the edit menu and then find the show contour map button checkbox here. Once you check it, it will show a contour map based on the gene expression level for the cells. And also this contour map is also adjustable in that you can change some of the parameters to change how the contour map is calculated. So, and finally, we offer to compare the gene expression level of a given gene across different Seurat clusters. In order to do that, you can switch to the summary tab over here. So now I will switch to the summary tab. It says it will now take a couple of seconds to load up this gene expression, EGFR gene expression comparison across different clusters from this experiment. Now it shows a violin plot. The horizontal axis is the EGFR gene expression, and then each of the violin represent the cells from that Seurat cluster, and then we can see that some of the clusters definitely have a higher EGFR expression compared to the other clusters. Now, I would like to talk about the ways to explore the differential gene expression available from the single-cell RNA-Seq, as both Zhenyu and Bill has introduced in the data production. So I will click on the differential expression tab, and this prompt explains that this will allow users to look at the significantly differentially expressed genes for one cluster compared to the rest of the cells. So a user should use this drop box to select any of the cluster. And so I will select cluster 0. It will take a few seconds to load up this view. Now, by default, it shows a table of differentially expressed gene from cluster 0 compared to all the rest of the cells from this experiment. Each row is one gene, and then there is this bar plot to show the log to fold change as well as adjusted p-value from that. I can scroll down this table to the bottom, and I can find out these genes are all ranked in descending order of log to FC, and then bottom genes, they are all have a negative log to fold change. At this point, I can click on any of these, any row, any gene from this table. It will now perform the gene expression. It will retrieve the gene expression of a VEGF-A, the gene I just clicked from the differential expression table, and then have it overlaid on the UMAP plot, just like what we already demoed. So I will go back to the differential expression. The last thing I would like to demo is the gene set enrichment analysis, which is available from this second tab next to the differentially expressed genes. So I will switch to this tab, and this analysis will be performed based on the genes that are based on the differentially expressed genes that are already retrieved for the cluster 0 that are in this table. So go to the GSEA tab. In order to run the analysis, in order to perform a very quick GSEA analysis, I just need to select a gene set group from the last drop-down menu. So here I will select the hallmark gene sets. So now this is the GSA results for each of the hallmark gene sets that shows a from the GSA analysis, where each row is one gene set, and then I can click on that gene set to show the GSA plot. And each gene set will also have a bar plot to show its normalized enrichment score, the gene set size, p-value, FDR, and then the leading edge of the genes from this gene set. So that concludes my app demo. I will be happy to, I think I will turn back to Sharon.
Sure, I can go ahead and share my screen now. Thank you so
great.
much, Jen.
Thanks, Jen.
Yeah.
And now we're going to get into the API demonstration portion. At the left here, we have some slides, and on the right, we have the terminal, which I'll use to demonstrate the single-cell RNA API. So just to give a little bit of introduction information, this API is what drives the single-cell RNA-Seq tool. This is accessible directly by end users, so anyone can access it. And it pulls expression data from the matrix layer of the HDF5 file. So if you open up the HDF5 file, that's where you can find the information that you'll find here. All data that you see here will be open access. As Zenu showed, everything except for the BAM files, which we won't be looking at, are open access. And we will be using curl in this demo through a Mac command line. But many other softwares will work. I know other people can use different browsers, or I know a lot of users use Python to access the GDC API. So all those are valid options, but and all of the parameters are pretty easily transferable. So let's talk about how it can be used. So the URL for the API is api.gdc.cancer.gov. And the first thing we can actually do is curl the status endpoint, api.gdc.cancer.gov status, just to make sure everything's up and running. I'm sure it will be. So right here, we know that the API is working. We will clear the screen. And the URL is, instead of I just used status before, it's after the main URL, you add single cell or scRNA underscore SEQ / and then gene underscore expression. So the input that we're going to give to this are the gene IDs in ensemble format, a case ID or a file ID, which will be in a UUID format, and the output will be gene expression values by cell ID. So let's start with a quick goal of retrieving TP53 expression data for this particular case from CPTAC3. So this here is what we call a submitter ID or a barcode in the GDC. But the GDC assigns everything a unique identifier. So here is the unique identifier for this case. And TP53, its ensemble ID is this one right here. So we'll need those both. And then if we look at the payload1.json, which is the file that we're going to be using as our input, you'll see that we have the case ID as one key, as a UUID, and then the gene IDs as another key, and that's the ensemble ID. And notice there's an S at the end. That's because this is a list format, and this has the option to add a few things, or more than one gene. So we'll get out of that, and let's go through and run this command. So we'll do curl. The request type is going to be a post. We'll use a header to tell it that we will be passing a JSON here. the data will be that file we just looked at, payload1.json. And we'll pass it to the UUID, or the, URL. Okay. And once we do that, we're going to see kind of a messy JSON output here. And a way to mitigate that is to use a JSON formatter or pipe it into a file and use a JSON formatter later. So we'll give this just a second to run the query. And here we go. You see this, it's pretty messy. So I'm gonna run that one more time, and we're going to use JQ as a JSON formatter, and then we're going to pipe that into a file called output demo.json. And we'll just give it a few seconds. And if we look at output demo, capital D demo.json, you'll see the output format here. It's pretty apparent. The case ID, the file ID, and the gene ID. So the case ID and the file ID is just a unique identifier for the file. And it belongs to this particular case. So this file came from this patient. And the gene ID, of course, is labeled too. And then each of the cells has a small section here that gives a cell ID followed by the gene expression value, which is a number. And as you can see, there's a lot of them, and we could go down for a while there. So I'll get out of this and clear the screen. And the next goal here, which I'm going to use to demonstrate having more than one gene, is to retrieve the expression data for EGFR and PTEN for that same case. So payload2.json has the same case, but instead of having one gene ID, it has two. So this is, two different ensemble IDs that we're going to pass to the API. So we'll rename this output demo 2, and we're doing everything the same. I'm just going to use payload 2 instead of payload 1. I'll go ahead and run that. This should take just a few seconds. So now when we do see it here, and remember that one was formatted, so it will look better when I open it up. Output underscore demo 2. We have the case ID, file ID, and gene ID, followed by a whole bunch of cells. And then after that will be kind of the identical object that starts here, but for the same case and file, but a different gene. So actually, if we search for gene ID, scrolling will take quite a while. We'll see the top one from the top of the file there, and then the next one a little bit down, and then that returns us to the top. So it's each of these genes has a specific object that has the same values in it. So that's it for the demo, but I'd like to introduce you to some more resources we have for documentation and some other things that are that we have going on at the GDC. So first, if you're interested in looking at the single cell RNA-seq tool, and which has some very in-depth documentation, you would go to right here where gdc.cancer.gov. You could go to portal.gdc.cancer.gov. Either one is fine. And the way to get to the documentation site is to go to the GDC apps button and click on documentation. Alternatively, you could just go to docs.gdc.cancer.gov. Now, to get to the single cell RNA-seq tool, you would go to Data Portal, and every app, every tool that you see in the GDC Data Portal, you will find a section about on the left here. So let's scroll down to single cell RNA visualization. And here is a lot of information about how this tool runs. If you're interested in the single cell RNA-seq API, you would click, of course, on API on the top toolbar here. And then on the left side, click on data analysis. This is where all the analysis endpoints live. And then on the right, which is like the table of contents for this particular page, you would click on single cell RNA-seq here. And this will bring you to a bunch of the information that I just gave you earlier in this presentation. But here you can copy and paste. Now, outside of the single cell RNA-seq, I want to point out a few other things that the GDC has available. So let's go back to the GDC website. So we'll just type it in gdc.cancer.gov. You can get to that from the apps menu too, but that's an easy way to get there. And if you're interested in the webinars, you would just go to support. And on the left here under training, you'll see GDC webinars. And here's today's webinar, but all of the past webinars should have YouTube videos. This one will be coming soon. And you can watch the past ones, including the single cell RNA-seq one from the past, which should be in September 2021 right here. So if you're interested in that, here is that one. Or, look for any other GDC topic you're interested in. We have lots of information about lots of things in these webinars. Lastly, if you're interested in things that the GDC is doing, like new releases, new software releases, new data sets that are coming out, I'd recommend going to support and click on join the GDC user listserv. It just takes a few seconds and you will get a notification about whenever the GDC does a new release of data, software, or anything like that. And it's usually once or twice a month you'll get something. So it won't spam up your email inbox. So anyways, I thank you all so much for joining. And if there are any questions, we'll be happy to take them.
Okay, it looks like there are no questions. So thanks so much, everybody, for attending today's webinar, and we'll get the video recording posted on the website as soon as we can. Thank you very much, and thanks to our speakers, Bill, Shin, and Zhen Yu. Have a great day, everybody.
Thanks all. See you later.